All right, today we are going to unlock an entire invisible world. A world that exists right on your skin, in a single drop of water, and, well, inside every living thing. We're doing a deep dive into the microscope, the tool that pretty much made modern biology possible. So if you've ever wondered how on earth we can see something as tiny as a virus, or more importantly, what you really need to know for your exams, you are in exactly the right place.
So, here's our game plan. First, we'll take a quick trip back in time to see how this all kicked off. Then, we'll get into the two golden rules you have to know, magnification and resolution. After that, it's the main event, a showdown between the light and the electron microscope. Next up, we're going to tackle the one thing you just can't escape, the magnification formula. And I'll make sure you can absolutely nail it every time. And to finish, we'll zoom back out and see why all of this is so much more than just a topic in a textbook.
Okay, part one, our journey into the unseen. Before we get bogged down in all the tech, let's just appreciate what a massive breakthrough this was. I mean, let's go back to a time when this entire microscopic world was basically just science fiction.
Seriously, just think about it for a second. A single drop of pond water is like a bustling city, teeming with life. Your own body is made of trillions of individual living cells. For pretty much all of human history, this entire universe was completely, totally hidden from us. So what was the key that finally unlocked that door?
Well, it all started with some very curious people. Back in 1665, a guy named Robert Hooke pointed a really basic microscope at a sliver of cork. He saw these tiny box-like structures that reminded him of the rooms in a monastery, which were called cells. And yeah, the name just stuck. A little while later, Anton van Leeuwenhoek was totally blown away when he saw tiny little animalcules, what we now know are single-celled organisms swimming around in water. But the real game changer? That came in the 1930s with the invention of the electron microscope. That was the leap that took us from just seeing cells to exploring the incredible machinery working inside them. And at the end of the day, all of these mind-blowing discoveries came down to this one instrument, a microscope. At its very core is a tool for making things bigger. It takes a hidden world, one that's way too small for our eyes to process, and it blows it up, bringing it into focus so we can actually study and understand it.
All right, section 2, the two rules of seeing. Now, to really get how microscope works, you've got to understand its two most important features. You can kind of think of them as the vital specs that define just how powerful it is: magnification and resolution.
Okay, first up, magnification. This one is pretty much exactly what it sounds like. It's just the measure of how much bigger the microscope makes something look compared to its actual size. So if the magnification is, say, times 100, it just means the image you're seeing is 100 times bigger than the real thing. It's as simple as that.
But you know, just making something bigger isn't the whole story. What's the point of a massive image if it's just one giant blurry mess? And that's where our second rule, resolution, comes in. This is absolutely critical. Resolution is all about clarity. It's the power to see 2 points that are incredibly close together as two separate points, instead of just a single fuzzy blob. The higher the resolution, the sharper and more detailed your image is going to be.
And this comparison here just nails the difference. See, high magnification without good resolution just gives you a bigger blur. But with high resolution, you can see all the fine, intricate details. You need both to really see what's going on. If you want to see the tiny little organelles inside a cell, you need high magnification to make them big enough, but you need high resolution to actually tell them apart. So always remember, magnification makes it bigger. Resolution makes it clearer.
Okay, so before we dive into the different types of microscopes or try to tackle any maths, we have to get on the same page. We need to speak the right language. And in microscopy, that means getting your units down. In this tiny world, things like meters and centimeters are just ridiculously huge. We need a much, much smaller scale. And trust me, getting these conversions right is absolutely non-negotiable for your exams.
So our first key unit is the micrometer. You'll see it written with this Greek letter mu, which, by the way, is pronounced mu. As you can see, there are 1,000 of these micrometers in one single millimeter. Just to give you a sense of scale, a typical animal cell is somewhere between 10 and 30 micrometers wide. This is really the bread and butter unit for cell biology. And if we want to go even smaller, we use the nanometer, or nm. Now get this, there are 1,000 nanometers in just 1 micrometer. This is the kind of scale we're talking about when we measure insanely small things, like a virus or the thickness of a cell membrane.
So keep this unit in your back pocket, because it's going to be super important when we start talking about the limits of resolution. Okay, part 3, light versus electron. Now that we've got the basics down, let's look at the machines themselves. It's time for the big comparison between the two main types you need to know. The light microscope, that's the trusty workhorse you'll find in your school lab, and the electron microscope, which is the high-tech powerhouse that drives cutting-edge research.
Let's start with the one you've probably already met, the light microscope. This is the classic piece of kit you've almost certainly used in class, and it's perfect for taking your first look at things like onion cells, or maybe even some cells from your own cheek. So, how does it work? Well, it's pretty straightforward, really. It shines visible light through a specimen and then through a series of glass lenses, which bend that light to magnify the image. It tops out at a magnification of around times 2000, and its resolution is limited to about 200 nanometers. But its advantages are huge for school science. It's pretty cheap, it's portable, and, this is the big one, you can use it to look at living things. You can literally watch a cell moving or dividing in real time, all in glorious natural color. But that 200 nanometer resolution limit, that's a hard wall. You just can't see anything smaller than that with light. So what if you need to? What if you need to see the tiny structures inside the cell's organelles? For that, you need a completely different beast. You need an electron microscope. Instead of a beam of light, this incredible machine shoots a beam of electrons. And because electrons have a way, way shorter wavelength than light, you can get unbelievably better resolution and magnification. Now, there are two main flavors of electron microscope you need to know. First, there's the transmission electron microscope, or TEM. This one works by firing electrons through an ultra-thin slice of your specimen. It's like an X-ray, giving you a flat, 2D cross-section, but with incredible detail of all the internal bits and pieces like mitochondria and ribosomes. Then you have the scanning electron microscope, or SEM. This one does the opposite. It bounces electrons off the surface of an object. The result is these amazing detailed 3D images that really show you the texture and shape of the surface. This table right here is the ultimate revision tool. It puts everything we've just talked about side by side. I mean, just look at the staggering difference in power. A light microscope maxes out at times 2000 magnification. An electron microscope can go up to 1,000,000. And the resolution, from 200 nanometers with light down to 0.1 nanometers with a TEM. But there are always trade-offs, right? To use an electron microscope, your specimen has to be dead, sliced incredibly thin, and placed in a vacuum. The images are always black and white to begin with. Oh, and the machines themselves are enormous and cost a fortune. Okay, section 4, let's get practical. Let's talk about mastering the magnification formula. I can almost guarantee you're going to get a calculation question on this in your exam. So let's break it down step by step so you can walk in there feeling confident you can get full marks every single time.
Here it is. This is the one you absolutely positively have to memorize. Magnification equals the image size, that's how big it looks on the page or in the eyepiece, divided by the actual size, so it's real-life size.
Now, here's where the examiners like to be a bit sneaky. They won't always ask you to find the magnification. They might give you the magnification and the image size and ask you for the actual size. That means you have to be comfortable flipping this formula around. A formula triangle is your best friend here. Just cover up the thing you want to find with your finger, and the triangle tells you exactly what to do with the other two.
Honestly, to solve any of these problems, just follow this simple 3-step recipe. Step 1, and this is the one that catches so many people out, check your units. If your image size is in millimeters and the actual size is in micrometers, you have to convert one of them so they match. If you don't, you'll get the answer wrong. Guaranteed. Step 2, write down the correct version of the formula you need. And step 3, just plug in your numbers and solve. Right, let's do one together. This is a pretty typical exam question. An image of a cell is 50 millimeters wide. It's been magnified by times 500. The question is, what is the actual size of the cell? Okay, let's use our 3-step method.
Okay, step 1, units. The image size is in millimeters and magnification is just a number, so we're okay to start. Step 2, the formula. We're looking for the actual size, so we need to rearrange our formula to actual size equals image size divided by magnification. Now we can substitute our numbers. That's 50 millimeters divided by 500.
Pop that into a calculator, and you get 0.1 millimeters. Now hold on, this is where a lot of people would just stop and move on, and they'd lose a mark. In biology, we don't really measure single cells in millimeters. The number's just too awkward. The question will often want you to give your answer in a more suitable unit, like micrometers. So we've got one last, super important step.
So, just like we said earlier, there are 1,000 micrometers in every millimeter. That means to go from millimeters to micrometers, we multiply by 1,000. So we take our 0.1 millimeters, multiply by 1,000, and that gives us 100 micrometers. See? A much nicer, more sensible number for the size of a cell.
And there we have it, our final answer. By following those steps, nice and methodical, we've confidently worked out that the actual size of the cell is 100 micrometers. It's that systematic approach that stops you from making silly mistakes and gets you all the marks.
Right, final section. Why does any of this actually matter? We've gone through the history, the theory, the tech, and the maths. But let's bring it all back home. Because microscopes aren't just for looking at onion cells in a classroom. They are absolutely fundamental tools that have shaped our entire modern world. I mean, their impact is literally everywhere. Think about it. In hospitals, doctors use them every single day to diagnose diseases by spotting abnormal cells. In forensics, they're used to examine tiny fibers or tissues from a crime scene. Microbiologists used incredibly powerful electron microscopes to actually see the COVID-19 virus, which was a vital first step in making the vaccines. And engineers are even using them to design brand new materials, one atom at a time. So, let's wrap this up. What are the key things you need to walk away with for your revision? Number 1, know the definitions of magnification and resolution and be able to explain the difference. 2, be able to compare light and electron microscopes, their pros, their cons, the whole shebang. 3, be confident using and rearranging that magnification formula, and for goodness sake, always check your units. And 4, understand that higher resolution is the real secret to seeing more detail. If you can do those 4 things, you're in fantastic shape. You know, from Robert Hooke's first little glimpse of a quark cell to modern microscopes that can literally see individual atoms, this journey into the world of the small has completely changed how we see everything. Which kind of leaves us with one last big question. Now that we can see right down to the building blocks of matter, what's the next invisible world out there that we still need to explore? And what new tool are we going to have to invent to finally see it?